Okay, good afternoon, everyone, and welcome to the closing keynote session at On Helix 2020. I'm Tony Jones, and I'm Chief Executive of One Nucleus, and it's been a pleasure to have your company for the past 2 days. And now, as we come to the closing session, I'm delighted we have three great speakers lined up for this closing session, just to really show us where innovation is going. And the title of the session, I think, was Sustainability, which I think we'll all see that just through being more effective, which we may come back to later. A couple of housekeeping notices just before we start. If you have questions for the speakers, then please do message Aline Charpentier through the network app for the conference, and Aline will feed them to me, and if time permits, then I'll put them to the speakers at the end of their particular talks. So without more of ado from me, I'd like to welcome our first speaker to the virtual stage, Alejandra Salas from Abcam, who's their Vice President of Product Development. Having joined Abcam in 2013, Alejandra now looks after the output from three major sites across UK, US, and China, and really is leading their innovation on some of the new product development. She's led several key initiatives, and previous to joining Abcam, she was R&D Director at Merck Millipore, and with studies at UCSF, Trudeau Institute, and UCL. So you've seen the global world. I'm looking forward to some global insights. Alejandra, over to you.
Okay, thank you very much. Can I have the first slide, please? The title of my talk is Supporting the Life Science Industry with Sustainable, Validated, and Consistent Reagents. It is my pleasure to be here today to discuss how Abcam is having an impact in life sciences by providing these sustainable, validated, and consistent reagents, as well as to show how these reagents are making their way into diagnostic tests and into the clinic. As Tony mentioned, my team focuses on innovation and constant process improvement, new product development to bring the best products to market. And this is integral to Abcam's ambition to support scientists at all stages of their careers so that they can achieve their next breakthrough faster. And in addition to having an impact in science, something that always keeps me excited at Abcam is our commitment to increase scientific reproducibility. And this is being taken into multiple activities that we have done globally, and our recent organized conference on antibiotic reproducibility a couple of weeks ago. But today I will share with you some of the key initiatives that are supporting reproducibility and how this can accelerate the development of diagnostics and therapeutics. Next slide, please. Abcam serves the healthcare and life sciences sector. And for any of you who don't know Abcam, we are a global life sciences company that identifies, develops, and distributes high-quality biological reagents, proteomics, and cellular tools. And we have over 1,400 employees in 12 different locations. Each year we serve over 600,000 scientists through our catalog of products, but also through custom projects. And we have developed over 3,000 custom projects to date. We provide products and services that enable research and drug discovery for in vitro diagnostics and therapeutic application. And we influence scientists that are from very early in their careers in academia, all the way to people bringing the next drug to market. Next slide, please. And Abcam's history is not new. We have been disrupting and innovating in life sciences for over 20 years. Although initially Abcam's model was more of an OEM supplier, our approach at innovating has changed quite a bit over time. And in the past 6 years, we have been investing in building our own products to be more proprietary in what we are offering, because we believe that Abcam's own products are doing a better job at increasing our impact with life sciences much more than anything else that we can find out there in the market. And with this shift, we have been focused on expanding not only the breadth of our offering, but the depth with targeted partnerships and strategic acquisitions, which have enhanced our capabilities, as well as we are constantly trying to remove constraints from our internal development. And this has resulted in several key acquisitions, which have strengthened our position on recombinant antibody discovery and manufacturing, such as Epitomics in 2012, Axiomics 2015, and Alico in 2019. And they have increased to our access to deep expertise and best-in-class platforms for recombinant monoclonal antibody discovery, phage display, and B-cell cloning, respectively. These acquisitions and partnerships have strengthened our position on recombinant antibody discovery and manufacturing, as I mentioned. But we still, even though we are now producing great specific antibodies, another area that we needed to really focus was antibody validation. And this is another area of constraints that we wanted to remove. And for the past 5 years, we have been leading the charge in the industry, utilizing CRISPR knockout editing to validate our antibodies. And this has led to the most recent acquisition of applied stem cell. And now having this in-house knockout cell line editing capability has strongly supported our antibody validation initiative, which is the largest across the life science industry. So basically, our internal capabilities now allow to faster develop high-quality antibodies and also allows superior antibody validation. Next slide, please. And with highly specific and consistent antibodies at the core of our reagent development capabilities, our new sources of innovation for outcome have been to access other ways to generate amino acids for single-plex and multiplexing applications, as well as strengthening our antibody conjugation and labeling capabilities to address industry demand for conjugation-ready solutions. And we are combining our existing antibody and protein-based offerings, and these capabilities are really expanding our portfolio and creating new added value products which support diagnostics and therapeutic development. Next slide, please. And this, we know these new strategies and technologies rely on an essential element, which is sustainable, validated, and consistent antibodies. And as we mentioned, cellular editing has been pivotal in this strategy, an integral part of our effort to support validation and scientific reproducibility. And in order to ensure the consistency of our reagents, we have focused our internal new product development exclusively on the discovery and development of the recombinant antibodies. Next slide, please. And here, generating high-quality antibodies requires effective strategies for immunity and design and protein expression, as well as antibody discovery and manufacturing, as well as specificity validation. And a key constraint for us in the past had been sourcing high-quality proteins from external providers until we actually brought that capability in-house, and now we can express high-quality recombinant proteins, which has allowed us to have end-to-end control of the entire process and also improve our batch-to-batch consistency. Next slide, please. And we know that the choice of antibody format combined with high standards in manufacturing has a great impact on specificity and consistency. Therefore, as I mentioned before, we are concentrating exclusively on the development of new recombinant monoclonals. And we have several approaches that we deploy depending on the target of interest. And one of these is um, for example, to hybridoma conversion or to hybridoma conversion to recombinant, B-cell cloning, NGS, or sepsis play. And all of these features result in rabbit monoclonal antibodies that are specific and very sensitive to the molecules of interest. Slide 8, please. And when we compare it to other common formats of antibody formats such as polyclonals or standard hybridoma-produced antibodies, recombinant rabbit monoclonals really have superior advantages, and they have improved consistency and reproducibility, as well as highest affinity and specificity. And since they are produced recombinantly, they are also fully renewable with high level of consistency, avoiding gene loss mutations and cell line drift, which sometimes can arise when hybridoma-derived monoclonals. In terms of antibody selection, a recombinant antibody is really the most robust format available. Next slide, please. So we know that traditionally polyclonal antibodies have been developed in rabbits, and particularly when the desired antibody is against a small molecule or post-translational modification, like in this case, in the case of a histone modification. This has created the belief that a polyclonal antibody is better for histone mods or other DTMs. And although rabbits are in general better than rodents are distinguishing subtle differences, such as post-translational modifications, the fact that they are polyclonals also comes with concerns, including potential cross-reactivity and target binding, as well as high batch-to-batch variability. And next slide, please. As you can see here, we demonstrate, as you can see in the left panel, it is challenging to achieve batch-to-batch consistency when developing polyclonals. And this, the issue here is that once the existing serum where that antibody was obtained finishes, you have to go back to an immunization and essentially generate a new antibody mix. In contrary, as you can see in the right, when you have a recombinant rabbit monoclonal, once you have identified a clone that has the desired specificity and lock of target binding, then you can expect the same performance batch to batch. Next slide, please. But irrespectively of the antibody format, all antibodies require in-depth characterization, and in order to ensure that they only bind to the target of interest, in the applications where people will be using them, we select the most appropriate application based on the particular protein targeted, and we use a combination of approaches to confirm the specificity. And this includes particular tissue types, particular cell lines that we know where people will be utilizing these antibodies, and including validation in the relevant tumor tissue in the case of PD-L1, as you can see here. As well as using the right positive control, we always try to identify negative control, which the target protein of interest is not expressed on. But for some particular targets, finding a negative control has been quite a challenge. Next slide, please. And this is one of the reasons why knockout models are key tools in the validation arsenal, and why it's now a standard part of our antibody validation process. And knockout models provide an excellent standard for validation, as they represent a true negative control, like it's called standard for negative control. And for the past 5 years, we have implemented a large-scale antibody validation using knockout models to verify the specificity of our antibodies. This has been done above and beyond the thorough characterization and validation approaches and multiple application testing that we have always done. Next slide, please. And We have taken a thorough approach validating multiple antibodies available to particular targets in knockouts. And here, as you can see, we have 5 antibodies to annexin A1, and this allows us to confidently identify antibodies that have specificity and also potential antibodies which may have target binding and are not fit for purpose. However, as you can see here in the last image, there is a polyclonal anti-annexin A1 for which we actually find the target binding. And in this particular case, this antibody is removed from the catalog, and we contact all of the customers we have previously purchased. So during this initiative, next slide, sorry. During this initiative, we have found that a significant number of top-cited antibodies show off-target effects, and this is obviously quite worrying, but not necessarily a huge surprise, since we know that there has been an issue with irreproducibility attributed to antibodies. So that's why we believe this initiative is key. And we recommend that when you use antibodies for your research, you go for antibodies for which there is confirmed specificity in multiple applications, ideally the ones that have been validated in knockout models. And making and testing knockout cell lines is a body of work in its own right. So to further support the life science research community, we have made available our range of knockout cell lines and lysets, available off shelf and validated and ready to use. So for the last part of my talk, I would just like to switch gears to explain to you how we are taking our reagents beyond basic research and into more commercial arena, into instrumentation industry and clinical partnerships. And so, as I have mentioned before, next slide please, we have discussed all of the different elements that we have implemented in our in-house developed reagents. And the central to this strategy has been to remove constraints to strengthen our recombinant antibody development process from beginning to end. And we have already, as I mentioned, increased our expertise in recombinant proteins, antibody discovery and development. We have also increased and de-risked our new product development and manufacturing capacity, and established antibody discovery centers of excellence in multiple locations around the world, and eliminated constraints by embedding internal in-house capabilities for cellular evidence. Next slide, please. All of these individual strategies, as I mentioned, are enabling long-term supply of antibodies, and the fact that this content is proprietary means that we have control over the IP, and we can customize the content as needed. And this has opened the doors to many collaborations and downstream opportunities. And as you can see here, we are now leveraging our antibody content to support instrument platforms, specifically, particularly for platform instrumentation platforms that enable multiplexing. And this is obviously extremely important, because when you can multiplex and detect multiple analytes in a small valuable clinical sample, this could be critical for detection both in tissue and liquid biopsies. Next slide, please. And so it's very exciting for us to see the work that we have done potentially many years ago, developing an antibody which is now in clinical partnership and at a stage where they are impacting patient outcomes. And there are a couple of examples that I would like to highlight. The ARB7, for example, is a fascinating story in prostate cancer. This is a good a very good predictive marker for splice variant of ALV that it can be used as a predictive biomarker to evaluate the efficacy of patients with metastatic prostate cancer. And Epic Biosciences came to us when there was not a single antibody in the market specific for splice variant 7. And this is just one key example of many. And we are using best-in-class antibodies in industry-leading platforms to support the life science industry, which are allowing us to develop the medicines of tomorrow. For this reason, providing sustainable, validated, and consistent reagents is an absolute priority for us. And we are aware that our responsibility goes beyond simply making an antibody for research use, as our antibodies are also used as key components in diagnostics and clinical platforms that can have a positive impact directly on patients. And last slide, please. With this, I'd like to thank you for your time and attention today. And if you have any questions, I will have to answer, or please feel free to reach out. Thank you.
Alejandra, thank you very much for the insights you've shared. One quick question I have, you're clearly very passionate about increasing the reproducibility and validation of research results and targets. What's your biggest frustration in driving it faster and forward?
Possibly awareness. I think if we had more awareness and if we had more market pressure, this could go faster. But it is also an expensive endeavor. So I think this is one of the reasons it goes slow. I think at the same time, training for researchers, new researchers coming into science is a big key area. I think if we all, this has to be a very, very large collaborative effort really for this to work. Founders need to be pushing for the use of renewable, sustainable reagents. The publishers need to be pushing for all of the information to come in together, as well as having well-validated reagents. And the scientists themselves need to know which formats are better for them. And I think it's a lot, a combination of factors, but obviously suppliers need to put their part big time as well.
Thank you very much. I think in the interest of time, we will move on to our second speaker. But thank you very much, Alexander. Okay, it's my pleasure now to introduce the second speaker of the session. It's Steve Anderson, who's president at Covance and CSO of Covance Drug Discovery. Steve has spoken at our events before and has a real passion about biomarkers and precision medicine, and actually his research interests align with that molecular pathology and oncology. But if I recall correctly, Steve is the first, certainly one of the first, if not the first, people to actually take a companion diagnostic through full development. So in that sense, I would welcome Steve's insights. I'm looking forward to his talk about the challenges in precision medicine. Steve.
Thanks, Tony. Thanks for the opportunity to speak at this virtual conference. And as Tony mentioned, I've got a passion around biomarkers. I'll talk about biomarkers today and the value and the importance of having an adequate biomarker strategy for sustainable success in drug development. We can move to the next slide. What I thought is, in addition to focusing on biomarkers and the importance of them, I wanted to add a couple of other topics, given that the last session was to talk about sustainability. And many speakers at this conference have touched on these as well in the current environment of the COVID-19 pandemic. And these include things like the evolving nature of trial design and execution, the role of decentralized trials, patient engagement, which are all rolled into that sustainable strategy and wrapped around biomarkers as a key element. So we'll talk about biomarkers and their role as a driver of precision medicine, the various utilities, and talk about how the complexity of the biomarker landscape fits in very well with the evolving clinical trial landscape. And at the end, I'll bring in some things that we're doing and that pharma and biotech is doing with regard to engagement of patients in trial design, execution, and to ensure retention of patients so that they get when the trials are done, examples of new innovative life-saving medicines. And then I'll finish by talking about some things that are really impacted by the COVID-19 pandemic. How do we ensure safety of patients who are enrolled in trials in this very, very unsettling time? And what's the role of decentralized trials in being able to do that? So let's move to the next slide. If we look at the importance of biomarkers in drug development, there are a variety of features that highlight that. Biomarkers tell us more and continue to tell us more about disease biology. They uncover targets for drug development, and they're relevant for success. And if we look in the left-hand slide, part of this slide, throughout the course of the drug development life cycle, the utility of biomarkers from moving from phase 1 to phase 2 to a registrational trial, and throughout the life cycle of development increases the likelihood of success. In the right-hand side of this slide, we see that the increase in the number of approved therapies in the US, FDA-approved therapies, in which biomarkers are a relevant part of the drug label. We've crossed over the 40% threshold, and that number continues to increase. And that's summarized on the next slide. And this was a published study that looked at the likelihood of success from first in human to a regulatory approval if there was an appropriate biomarker strategy employed versus not. And the likelihood of success is increased threefold with the incorporation of an appropriate biomarker strategy. Next slide, please. So biomarkers are relevant for a whole variety of reasons in the drug development life cycle. They're relevant for understanding mechanism of action. They're relevant for understanding pharmacodynamics and pharmacokinetics, for selecting patients for enrollment in a trial, for stratifying patients into the treatment arm versus standard of care. We're seeing evolution of resistance markers, whether they be in immuno-oncology or other targeted therapies in oncology. Prognostic biomarkers, which tell us the course of disease and are relevant also for understanding severity of disease. And we're seeing them being used as surrogate endpoints more and more. Next slide, please. So the relevance of biomarkers and the value that biomarkers provide, can be distilled into a number of features. So one, it reduces the average development cost and time. It was shown in earlier slides. It reduces the likelihood of an adverse event or a safety concern. It identifies patients who will best respond to a therapy. And all of those put together really highlight the fact that while you invest in dollars up front for the delivery of biomarkers into your strategy, the downstream impact is a cost-effective savings and a greater likelihood of success. Next slide, please. So as we look at the evolution of biomarkers in the era of precision medicine that we live in today, on the left-hand side, we see what's happening in the therapeutic landscape On the right-hand bang boxes, we see what's happening in the biomarker tools and applications. And in the middle, the bubbles also talk about the role of the combination of the two. So we see that trials are biomarker driven, and I'll focus mainly on oncology, but it's true now in other therapeutic areas. We see that the complexity of therapeutic development is increasing, whether it be cell-based therapies, adoptive cell therapies like CAR T, in which the therapy is a living cell extracted from the patient, engineered and reintroduced to the patient. So complexity of the types of therapies, complexities of the modalities of therapies, and so combination therapies being relevant. And if we look at the complexity of biomarkers, that also is playing into the current landscape. We're seeing the evolution of next generation sequencing technologies more and more, where Tony mentioned in companion diagnostics, we're seeing now a multiplex platform like the Foundation One assay from Foundation Medicine being applied as a companion diagnostic for a variety of new therapies. We see other multi-omic type approaches like digital pathology, digital spatial profiling, multi-proteomic based assays that tell us more about the biology of disease and link up very, very significantly with the drug development modalities as well. So if we move to the next slide, I think the role of biomarkers in understanding the therapeutic, the impact on the patient, and the delivery of precision or personalized medicine is very well illustrated today in biomarkers that are applied for adoptive cell therapies like CAR T. It's a very complex interaction between the unique aspect of the therapy as well as the target population of cells in the patient, as well as the role of the immune response in the patient. So we need a whole variety of tools to bring in to understand the aspects of both efficacy and safety of those therapies. We move to the next slide. What we're seeing in this space is the use of a whole variety of tools at one time to evaluate patient, to evaluate the efficacy of the therapy, its persistence, its expansion once it's introduced back into the patient. So it's pharmacokinetics, it's pharmacodynamics. Is it attacking the cells of importance, the cells in the B-cell lymphomas or B-cell malignancies? What is the safety profile? Is the immune response, is it a cytokine release storm that's being unleashed? Toxicology, so has the retroviral vector for the CAR T therapy been integrated inappropriately, causing a toxic profile versus a efficacy profile? So all of these things come into play. So the dose up front is the therapeutic activated and expanding in the patient. Is it persisting appropriately? Is it attacking the cells of interest? Can we look at the safety profile as well as the efficacy profile? And then also looking at whether or not this holistic approach leads to a better assessment of how patients are responding to these novel therapies. Let's move to the next slide. So just as the complexity of biomarker and a biomarker strategy is playing out, we also see this in trial design. And this slide, if we focus in oncology as a first example with other therapeutic areas to follow, for those of us who have been in this space long enough, we've seen the evolution from a chemotherapy approach on the left-hand side, where the time for the trials from first in human to success was often greater than 10 years. They followed a standard formula of dose escalation, choose the right dose, migrate that into a large patient population, very much a tiered approach and a separated approach. Phase 1, dosing strategy, phase 2, phase 3, registrational, and then leading to approval if efficacy is shown and safety is shown after that decade or longer period of time. We moved into the targeted therapy approach where there's many, many examples where the dosing strategy may be the same, but our understanding of efficacy starts to move back into the phase 1 trial, and the phase 1 and phase 2 designations blur, and we move into phase 2 in a selected population based upon the biomarker as the registrational part of the trial. And then post-commitment, post-approval, a commitment to expand the trial in a phase 3 trial. And that has taken the timelines down to 5 years or 8 years, or in some examples, from first in human to approval in about a 5-year window. When we move into immunotherapy, including the examples of adoptive cell therapy, the plans and the delivery are very different. So in immunotherapies like checkpoint inhibitors, you often have an adaptive trial design. You go from a phase 1, and that often can be expanded, and the phase 1 or phase 2 becomes the registrational element of the trial, with again an approval in less than 5 years. And so we've seen advancement in the way in which trials are designed and executed, in many ways driven by the lack of a targeted population to a very defined population. So if we move to the next slide, and that's illustrated now in some examples of platform trials, the basket trials and the umbrella trials. Umbrella, where you have a single tumor type like breast cancer and potentially multiple biomarkers linked to different therapeutics, and you look for whether or not there is a signal in any one of those combinations. And basket trials, where you have multiple tumor types in a single biomarker, like microsatellite instability for checkpoint inhibitors, and now tumor mutation burden. So again, complex designs, but biomarkers being an integral part of that. Next slide, please. So what's happened then as we've moved from a chemotherapeutic approach to a more targeted and immuno-oncology approach, the trial approach has really migrated as well to be much more streamlined. And instead of discrete phases, it's more of a learn and confirm approach. So in the learning phases, the early phases, does the drug work? What are the targets or the biomarkers? What is the appropriate dose? Is it safe? Can we predict response in those early dose escalation studies that translate into knowledge to be applied in the registrational trials? And can we apply a Bayesian design to collect data through the course of this seamless design and modify appropriately? The confirm, the later phases then are intended for the drug filings. Are we choosing the right endpoints? They may still be surrogates. And how do we apply an adaptive design to, again, achieve the endpoint? So again, learn and confirm versus more of a standard phased approach to drug development. Again, biomarkers being a relevant and a critical component of that trial design. Let's go to the next slide. And this slide, again, it shows how conceptually in oncology, but also in other therapeutic areas, we really have an iterative and integrated approach to drug development. And that involves from drug discovery through a registrational product. We learn as we go, and we apply that either in other phases or in our thinking around trial design. And with regard to oncology, that is, we have the appropriate preclinical models. We're moving to things like organoids and organ on a chip to help understand and supplement what we do with animal models, biomarkers and companion diagnostics still being extremely relevant, and again, incorporating those into the appropriate trial design. But rather than that linear process, now it's a very integrated and iterative process. As we think about that, moving to the next slide, it's also time for us to think about how we engage the patient better, how we leverage new tools to collect information appropriately from patients to ensure that they're engaged, and also the retention of patients through the course of a trial is enhanced. Patient centricity is something that's been floated around by a number of groups and organizations, and it really is appropriate that we start to think of the patient and rethink and redefine the patient as the center of drug development and as the center for the development of diagnostics. It's critical for adequate trial delivery. We're seeing this more and more as patients own more of their own health decisions. It's not a singular activity. Patient centricity doesn't mean one thing. There are multiple factors that play in. It really is how we engage patients, how we use data from that feedback from patients to design trials, to keep them engaged, to execute on trials better, and thereby to bring novel therapies to patients faster and in a better way. So I'll move to the next slide. And on the next slide, this is sort of a journey map, very simplistic, but I think looks at the holistic approach of engaging patients. We've taken on a role working with a lot of our pharma and biotech sponsors and leveraging patients who are part of our diagnostic division to understand what's important for them. What will they do and what won't they do as a participant in a trial? And we're seeing that in multiple areas. We're seeing that right now as we poll patients around their interest to be involved in novel therapeutics to address the SARS-CoV-2 virus. We leverage patient insights, patient profiling through surveys, through engagement. We also then share that information with sponsors, with sites, with coordinators at the sites. So again, we understand the impactfulness and the thinking of all of the relevant parties in the drug development process. Through the course of a trial, we continue to engage patients around how they're feeling, what they think is relevant for the trial design, what is burdensome to them, what is working well, what is not working well. Provide that feedback back to the trial sponsors, to the sites, and then by doing so, by collecting that information, there's a greater likelihood that patients will be engaged along in this process. They'll be retained in trials, and that will lead to greater success. We move to the next slide. As we've been thinking about this, it's it's data-driven insights and engagements that are relevant. So we look at those actionable insights. It's the voice of the patient, it's the voice of the sponsor, it's the voice of the investigative sites. It's what we can do to leverage data to target patients that would be recruited into a trial. Again, whether that's an oncology trial or a COVID-19 vaccine trial. Decentralized trials, as I'll talk about in a minute, will become a relevant part of that and are becoming a relevant part of that. Therapeutic area expertise is relevant. As we look at optimal study, again, it involves input from patients, sponsors, and sites, and the data that we collect now in applying new tools, wearables, artificial intelligence to collate a lot of that data, again, relevant for the patient as the center of our thinking. Next slide, please. So as an organization, I think as an industry, there's been a movement to think about, especially driven by the COVID-19 pandemic, how do we keep patients enrolled in trials in a safe manner? And how do we do that leveraging new tools and technologies? And I think we're moving down this continuum from the old way of doing things, where it's a full site engagement, where patients go to the investigative site for all of their relevant visits, for all of their relevant lab work, for all of their relevant assessments. Historically, a virtual model has applied more to an observational study or to a post-marketing study, but we're now starting to see a blurring between these lines. And how do we leverage tools, processes, et cetera, to close that gap, and where appropriate, leverage that decentralized trial approach.
Just coming
Next
up on
slide. Okay, I got just 2 slides. So again, I think I'll skip this slide and move to the last slide. The only thing I'll say on this slide is that it's a collection of important elements that are relevant for the delivery of decentralized trials. Move to the next slide. And again, it has to be a way in which we design around the patient as the center, how we engage sites appropriately in their engagement with the patients and with technology and tools. Let's go to my last slide and I'll finish up. So drug development is increasingly dependent upon an appropriate biomarker strategy. It's impactful for success, and the complexity of biomarker analysis is becoming an everyday occurrence. Trial designs continue to evolve and have to be overlaid with biomarker strategy. They're adaptive, they're iterative. The learn and confirm approach is becoming more of a standard way of thinking of trials versus the linear approach. And patient-centric approaches will be critical, not only for today, but sustained success in drug development. It's important to connect with the patients appropriately, to understand their voice in trial design, but also to poll them during the course of a trial about what's working, what's not working, in order to engage and retain them in the process. And then we're starting to see, driven by COVID-19, but other elements, an increasing focus on the role of decentralized trials. All 3 of these elements, I think, become critical for sustainable success in the drug development industry. And Tony, with that, I'll stop and see if there are questions.
Thanks, Steve. Very much appreciated. I'm just a little bit conscious of time as we're going out live, and people can contact Steve through the app directly with his follow-up questions. And I would encourage people, if they've got other more broad questions, also to use the social media and the hashtag of OnHelix20, because it's a very insightful talk. Steve, thank you for your time today.
Thanks.
And
thanks, Tony. Glad to engage that way.
Thank you. Okay, so we move to our final speaker of the OnHelix 2020 agenda. I'm delighted we have Akshay Vishnu from Alnylam Pharmaceuticals, who's the President of Research and Development. Joined Alnylam in 2006 and has subsequently now leads the discovery and development of their novel pipeline in RNAi therapeutics, including the recently approved Onpattro. So I think there's another exciting area coming through of how we're all doing this better. Then I'm delighted we have Akshay here to share his insights. Akshay, over to you.
Thank you, Tony. So I'm presuming I'll take about 15 minutes for the slides. Is that about right? I know you were running slightly late. Okay. All right. Well, hello, everybody. Good morning, good afternoon, good evening, wherever you are in the world. I hope everyone's had a good meeting. I've got to tell you, this is the first meeting in terms of a conference that I'm doing since the COVID period started, and I'm absolutely delighted to be doing it. The circumstances are unusual, but I'm glad that our community has risen to the challenge and we continue our science and communications as creatively as possible. So thank you, Tony, for the invitation and for this opportunity. With that, maybe we can start the slides. I can't see them. Are they up and ready? Yes, Tony? I can't hear you, Tony.
I'm waiting for the tech guys to say if they're up. I can't see them either.
Okay.
Yeah, they're up. Yeah.
Okay, so the first slide is the title slide, yes? Yeah, okay, great. So over the next 10 to 15 minutes, I'll give you a talk on the Alnylam platform, where we've been developing RNA interference, RNAi therapeutics over the last 18 years. And the platform over the last few years has come into its own as it's matured and really beginning to give us a very productive drug discovery and development engine that we hope to harness for many new applications beyond the one that Tony mentioned on Patro. So with that, let's go to the next slide. So the agenda is basically, I'll give you a brief introduction to RNAi and the journey from bench to bedside, and then focus in the second part on how we've actually built the pipeline, and then really a very exciting time frame right now where we see many, many future applications for the technology across a range of therapeutic areas. So next slide. So RNAi therapeutics, I imagine most of you are familiar with RNA interference, but this schematic is just to introduce you to it in case you aren't. RNA interference is an evolutionarily very old process. It's an oligonucleotide-driven process, and if we look at the schematic on the right-hand side, what you see is a small interfering RNA coming into a cell. That small double-stranded RNA is loaded into a molecular complex called RISC, and the antisense or red strand is retained within the RISC complex at step 2. Now, the sequence of that red antisense strand serves as a template to bind any complementary target messenger RNA that has the matching sequence, and that's what you see going on as a messenger RNA is bound. And once the antisense strand has bound its cognate or complementary messenger RNA, there's a cleavage that takes place of the messenger RNA. And once cleavage of messenger RNA occurs, of course, you can't make the protein associated with that mRNA, and we effectively dramatically downregulate the protein by virtue of this sequence-driven mechanism. The beauty of this is not just specificity, but also the potency, in that the red antisense strand is retained for further rounds of target cleavage within the RISC complex. And so one copy of an siRNA, once introduced into a cell, will target cleavage of hundreds of thousands of copies of the target messenger RNA. And next slide. Now, the story of RNAi really began almost 30 years ago when in 1990, Stefan Jurgensen and his colleagues discovered that the process was evident in flowers. And specifically, they reported the process they termed post-transcriptional gene silencing, subsequently recoined RNAi, but they were the first to describe it in flowers. And in Jurgensen's lab, they showed that essentially a small RNA could target degradation of an enzyme called chalcone synthetase that allows petunias to develop their purple color. And degradation of chalcone synthetase led to pale coloring of the flowers. And so you got these variegate or very white flowers. And this was thought to be an oddity just in plants, but subsequently, and very importantly, Fire and Mello in 1998 described a similar process occurring in worms in the model organism C. elegans, where they described gene-specific silencing driven by a small interfering RNA, and they coined the term RNA interference. And the founders of Alnylam by 2002 had described similar processes occurring in Drosophila and also in HeLa cells and other human cell types. And with that, Alnylam was founded in 2002 with the promise that an siRNA or small RNA-based silencing mechanism could silence any gene in the body, and with that, we could target pathogenic proteins that brought about disease, and by reducing their levels, hopefully lead to therapeutic benefit. Importantly, in 2006, the importance of RNAi was recognized by the Nobel Prize for Fire and Mello. And after a long journey, which I'll describe very briefly, we got to our first approved product in 2018, which we call Onpattro. Next slide. Now, at the core of our journey has been the following, that we knew that on the top left here, you see silencing of lamin in HeLa cells. This was one of the first papers by our scientific founders in the early 2000s. And we wanted to do that in a target organ against a genetically validated target. And as the last speaker said, with biomarker evidence of target knockdown, And in order to do that, we had to get our siRNA to that target organ. We had to deliver the siRNA to the liver. And it's that journey which, in at least 2 cases now, has led to approved products, Onpattro and very recently Givlari. And I'm going to talk a little bit more about that now. So next slide. This is just to say that we now have 7 drugs that are either approved or in the latter stages of approval. Onpattro and Givlari at the top here I've mentioned. Lumasiran, the third one here, is for another orphan disease. We recently got the phase 3 data. It's currently under review at the US and EU regulatory agencies. Inclisiran, some of you may have heard of. It completed phase 3 in hypercholesterolemia with the medicines company. We'd out-licensed it to the medicines company. Novartis then subsequently bought the product. It's under review at NDA. The NDA is under review with the US FDA, as is the MA with the EMA. And then there are multiple other programs in late-stage development. So next slide. And we've continued this push of drugs into the clinic once we matured the platform technology. And there are another 7 programs that are either in the clinic in phases 1 or 2 or about to be filed, including an siRNA against COVID, which we will deliver via the inhalational route, which we can talk about in the Q&A. Next slide. The journey, at a high level, one of the more notable features that's coming across now is that using a technology that's sequence-based with the specificity that we have, addressing genetically validated targets, biomarker-driven drug development. If you look at the right-hand side of this graph, you can see that the probability of success cumulatively for our programs is 58.4% when you go from the IND filing through to positive phase 3 data. And that's at least 5 to 10-fold better than the industry average. And so that's really taking advantage of a platform technology, focusing on an organ and a cell type that matters to us, and then focusing further on genetically validated targets, and then turning the handle and doing the same thing over and over again against different targets using a technology that's well understood and well established. And so we're really very excited by this number. And we obviously hope to, beat the industry average and continue numbers like this into the future. Let's see, but we certainly remain very optimistic. Next slide. Let's get into some of the details of how we built the pipeline that I just showed you. So, when the original description of RNAi came about in petunias and then C. elegans, Drosophila, and HeLa cells, everyone was very excited about potential applications to human disease, and Alnylam was started with great fanfare in 2002. However, there were a number of key technological challenges, not least that these are macromolecules. They're about 14 kilodaltons. They're hydrophilic. They don't go through cell membranes. They're easily degraded. And so there was an enormous kind of physicochemical barrier to getting our drugs to a target tissue where we wanted to address a genetically validated target, which in our case was the hepatocyte in the liver. And we undertook between 2002 and 2012 basically a decade of delivery research to figure out how to deliver an siRNA, our drug substance, or the API, to a hepatocyte. And next slide. The first of the solutions we reported in 2012 in the context of a human POC was the siRNA encapsulated, as shown top left, in a lipid nanoparticle. And the lipid nanoparticle is composed of multiple lipids, as indicated on the right-hand side through the bullets. Critical to this is that one of the lipids is an aminolipid, which is able to allow uptake by the hepatocyte by a mechanism which we've published on extensively and I can talk about, and then release via the endosomes, endosomal rupture into the hepatocyte cytosol, and the siRNA is then loaded into RISC and allows the RNAi program to execute. We had to go through several generations of lipid nanoparticles before we arrived at one that was safe and effective in animals that would carry through to humans. I'm going to now mention to you the story of Onpattro using a second generation lipid nanoparticle. Next slide. Now, Onpattro was developed for a disease called hATTR amyloidosis. This results because of a mutation in a gene uniquely expressed in the hepatocyte called transthyretin, or TTR. The mutant gene product comes out of the liver, misfolds, and deposits in nerves and causes a severe intractable neuropathy in middle age, which leads to death. And the severity of the neuropathy is measured by a scoring tool termed the modified neuropathy. On the left-hand side here, the mNIS+7 composite monitors the severity of the neuropathy, and then on the right-hand side, you can monitor the quality of life using the Norfolk quality of life tool. So what we did in early development, I'm obviously having to race through a lot here, is we showed that delivery of an siRNA against transthyretin in these patients led to reduction of transthyretin in the peripheral blood. So that's the key biomarker that demonstrated the proof of concept. And having demonstrated dramatic reduction of transthyretin in the peripheral blood of these patients, we then asked the question in phase 3 in a large randomized double-blind placebo-controlled study, what happens to the severity of the neuropathy and what happens to quality of life? And what you're seeing here is that in placebo, which is in red, the neuropathy worsens, the mNIS+7 score just inexorably worsens over time, as does the quality of life. Whereas in the Onpattro or patisiran-treated patients, you see an improvement, not just stabilization, but an improvement over time as the pathogenic protein levels are lowered in their blood with Onpattro treatment. And both from a neuropathy severity and from a quality of life viewpoint, they improve. So we didn't stop there. Next slide. We found another way to deliver an siRNA, and that's schematized on this slide here. So if you look top center, the siRNA is ligated via linker to a triantennary carbohydrate complex of GalNAc and acetylgalactosamine. These sugars are recognized top right by a receptor on the hepatocyte called the acetylglycoprotein receptor, or ASGPR. They allow the siRNA to be internalized and released into the cytosol, and then again an RNAi program to be executed. Now, the beauty of this mechanism is that it allowed, in contrast to Onpattro on the last slide, drugs developed by this means can be given subcutaneously. And we have gone on to develop, if we go to the next slide, all our drugs in this GalNAc subcutaneous conjugate format. We call them GalNAc conjugates. And patients can give themselves the drug, or a physician can give the drug. And on this slide, we're illustrating our second positive phase 3 program, givosiran. Now, this too is an orphan disease like hATTR amyloidosis. And what you can see here is that in this disease, which is termed acute intermittent porphyria, or rather acute hepatic porphyria, patients get recurrent attacks because of a mutation in a gene expressed in the liver. Downregulating that gene results in lowering of biomarkers that are mediators of the disease, specifically two chemicals termed aminolevulinic acid and porphobilinogen. In early development, we demonstrated dosing our drug in patients led to reductions in these pathological biomarkers in the peripheral blood. And then in later development, we demonstrated that lowering of ALA and PBG leads to reductions in the severe abdominal attacks these patients suffer with severe abdominal pain, back pain that leads to their hospitalization, not just once or twice, but we're talking 10 to 20 times a year in young women and men. And so this is a really dreadful disease. And we've been able to show in the phase 3 context in a randomized placebo-controlled setting that administration of givosiran, which is now approved as Givlari, results in a 90% reduction in the median composite attack rate. That was the primary endpoint. So we're delighted with that. And this drug, like Onpattro, is now approved in US and Europe. Let's just quickly wrap up with the future outlook. So next slide, and then the next slide. Growing safety database is the slide we should be on. We now feel very confident, having developed our first few drugs in rare diseases, that we can address much larger diseases and get to common disease. And illustrative of that, if we go to the next slide, is this drug inclisiran, which was developed in conjunction with the medicines company and now under the Novartis umbrella. These are phase 3 data showing injection of the GalNAc conjugate format of an siRNA against the target PCSK9, a very well genetically validated target for hypercholesterolemia. And you can see in the blue line relative to placebo, the LDL cholesterol levels dropped by 54%. Now, of greatest note here is if you look at the injections in the upper part of the diagram, you can see the injections are 0, 3, 9, and 15 months. In other words, this is a once every 6 month subcutaneous injection for this disease. And in a disease where patients, more often than not forget to take their tablets or forget to refill their prescriptions, this is a wonderful compliance advantage where they can be given their shot of the medicine by their GP or a nurse or in the local pharmacy, and we can really get the benefit of lowered LDL in the population. Next slide. We've developed a drug for hypertension, or rather we're in the process of developing a drug for hypertension. This too will be given once every 3 to 6 months. It's given against a target called angiotensinogen, a well-validated target for hypertension. Just recently, bottom left, we reported that in phase 1, we saw more than a 10 millimeter drop in blood pressure in patients treated with this drug at an HT relative to placebo. So again, like the last slide, we feel we can develop a subcutaneous therapeutic infrequently administered for a common disease, hypertension. And finally, to finish, we continue to develop the platform. And if we can just go through this is animated, maybe I shouldn't have animated it for this format. So if you click it 5 times, there should be 5 panels that pop up. Just to say, of all of these, the most important thing I want to emphasize is that the platform advances allow us to address new tissues. So all the work I showed you so far was to address genetically validated targets in the hepatocyte, in the liver. We've now developed delivery mechanisms to get our drugs to lower part of this slide in the middle, to the nervous system and the eye. And there are many toxic gain of function proteins in the nervous system and the eye, which if we can reduce them, will bring about, we think, a dramatic benefit. I mean, examples we're all familiar with are things like Huntington's disease, or alpha-synuclein in Parkinson's disease, etc. And so we're busy working on that part of our pipeline too, and hope to bring our first CNS drug to the clinic next year. And so with that last slide, if we just do all the clicks here, 1, 2, there are 3 clicks, just to say that having started the journey, spending about a decade on figuring out how to deliver our drugs, we're now accelerating the pipeline, not just towards liver targets, addressing rare disease, but also increasingly common diseases like hypercholesterolemia and hypertension, and new delivery advances that are allowing us to go and build pipelines for newer organs such as the nervous system and the eye. And I haven't even had a chance to update this slide because really I should put the lung on here as well, because we now have a COVID program, which is currently in the preclinical arena, and we're doing the CMC to bring it to the clinic, hopefully by the end of the year or so. So with that, I'll stop. But last slide, this is just to show you the flow of products that are now resulting from this rich platform that we feel we finally matured. It's been a fascinating and rewarding journey, but we think that we can make a real contribution moving forwards now. So thank you for your attention. I'd be happy to take questions.
Thank you, Akshay. It's interesting you touched upon your COVID work just at the end there. In terms of what you've seen from, you've brought the regulators a long way to compress those times with a novel platform over time. And you're going from rare to large disease. But on COVID now, with the way that's shaken up the way regulators are behaving, do you think that will translate back to the other processes you had to go through, and COVID will have changed the regulators for good, or will they revert back?
Well, that's of course a great question. In actual fact, it's not, from our perspective, what you just said, we've been hoping for, not just from the COVID angle, but even before that. If you think that our drug substance is the same every time, right? It's a string of nucleotides, G's, A's, T's, C's, let's say. Our drug substance is a very common program to program. The only thing that's different is the sequence to address each distinct mRNA. And so we feel that there's an argument to be made to truncate toxicology and manufacturing programs to support entry into the clinic so that they're leaner and more efficient. And so we hope to change regulators' mind on not doing a stereotypic, IND-enabling package every single time, but to allow some efficiencies and cost savings, as well as time efficiencies, to get these drugs to the clinic faster. Now that we've matured the platform the way we have, and it's becoming so much more predictable. Equally, one can imagine that once a product is approved, if there's a second generation that's the same sequence, then really one shouldn't have to go through the whole thing again, So we're actually teeing up those kinds of discussion independent of COVID with regulators, and we feel we have the arguments to support that.
Excellent. I'm conscious of time. Thank you very much for your time this afternoon.
Thank you.
Insights, and that's a great note to finish on, I think. Very optimistic. Thank you very much.
Thank you again for the invitation.
Thank you. So that brings us to the closing remarks of On Helix. And that falls to me to do a number of things. But I think more than anything over the past 2 days, it's really highlighted to me that we have complex ecosystems giving us complex collaborations at scale, and the industry response to COVID has certainly emphasized that and the dynamic. But we need that degree of complexity on our side to really address the complexity that we face challenges in any form of biomedical research and making progress. So it's been really interesting to hear the last 2 days, all the insights confirm for me that great stuff has gone on, great stuff is still going on, and there's even more great stuff to happen in the future. So I think I go away from this 2 days of conference feeling the future is quite bright. And I hope we see much of what's talked about today play out in the clinic. Just some thanks towards the end. Obviously, thanks to all our speakers over the last couple of days, both the ones who've been live and interactive, as well as the on-demand creating that content. We couldn't do it without them, and we couldn't do it without our sponsors who enable a lot of this. So thank you to them. And also, we can't do it without delegates participating. So thank you to all of you also. Last but not least, of course, there's another group that we couldn't do this without, and that's the On Helix team. I think COVID has shown us we've had to adapt. We work in one of the most innovative industries you could ever dream of. We need to be innovative when we don't want to be the weak link. So hopefully you found our transition to online and the way we've continued to evolve through this period has been supportive and it's delivered value for you also. And I'd just like to thank the team for the efforts they've put into that. And then I guess you thought it was all over, but not quite, honest. A couple of reminders: the app and the on-demand content, including recordings of the live sessions over these days, will be available for another 4 weeks yet for all delegates to access and to contact each other and remind themselves of maybe what they saw or maybe catch up on things they missed. And then finally, of course, looking forward to December, then we have our annual Genesis conference. It's a changing situation. We don't know quite yet what the situation will be in terms of physical gathering or virtual, but we are live and taking registrations and planning a virtual form of Genesis. And if the situation changes, that we can convert that to a form of hybrid and capture the best of both worlds, so people have the option to either choose to then convert to a physical delegate or remain as a virtual delegate and still engage in the conference, then we'll keep you updated. But if you go to genesisconference.com, the details so far are there, it's open, and we'll look forward to seeing many of you then. In the meantime, thanks for all your engagement over the last couple of days of On Helix, and we look forward to seeing you soon. Thank you.